Good morning, boys and girls. Today we are going to discussing an important concept is microsporogenesis. In that microsporogenesis, mostly we have discussed how to develop the pollen grains from the anther. You know that anther in previous classes already you have heard the structure of anther. First of all, what is meant by microsporogenesis? Here, the process of formations of microspores from microspore mother cell through meiosis division is called microsporogenesis. In that microsporogenesis, for the formations of the microspores, which divisions are required? Here, first of all, in angiospermic plants, two types of the anthers are present. First one is called dithecous anther, second one is called monothecous anther. This is one dithecous anther. In that dithecous anther, two lobes are present, two thecas are present. In each lobe, two microsporangium are present. Totally two lobes are present. In that two lobes, totally four microsporangium or four pollen sacs are present. Here, this is nothing but one dithecous anther. This is one microsporangium. This is one microsporangium. This is one microsporangium. This is one microsporangium. When the anther is young, a group of compactly arranged homogeneous cells are called sporogenous cells, occupy the center of each microsporangium. The ploidy of the sporogenous cells is diploid. You know that the sporogenous cells are developed from the primary sporogenous tissue. The primary sporogenous tissue developed from the archisporial cells. The archisporial cells are located each corners of the anther. The archisporial cells are located each corners of the anther. Right here, this is sporogenous cells. The sporogenous cells, it undergoes mitotic division to form two microspore mother cell or two pollen mother cells. The ploidy of the microspore mother cell is diploid. The microspore mother cell or pollen mother cells are developed from the sporogenous cells through the mitotic division. How many pollen mother cells are developed? Two pollen mother cells are developed. Next one. Actually, two pollen mother cells are developed. Here we have discussed only one pollen mother cell or one microspore mother cell. As the anther is matured, the microspore mother cell or pollen mother cell, it undergoes meiotic division to form pollen tetrad or microspore tetrad. The ploidy of the tetrad is haploid. The haploid condition is represented by the N. The haploid condition is represented by the N. Generally, in angiospermic plants, different types of the tetrads are formed. First one, this is nothing but tetrahedral tetrad. Tetrahedral tetrad is mostly present in dicotyledon plants. Second one is isobilateral tetrad. The isobilateral tetrads are mostly present in monocots. Third one is decussate tetrad. Decussate tetrad is present in Magnolia. Fourth one is T-shaped tetrad. It is mostly seen in Aristolochia. And the last one is linear shaped tetrad. The linear shaped tetrad is seen in Halophila. Among them, 5 types of the tetrads. The most common type of the tetrads is present in all angiospermic plant is tetrahedral tetrad. Among them, 5 types of the tetrads. Tetrahedral tetrad is commonly present in all angiospermic plants. Here, this is nothing but one tetrahedral tetrad. In the tetrahedral tetrad, here approximately 3 pollen grains are appear. Where is the one more pollen grain? The fourth pollen grain is located back side of the tetrad. Here at this stage, the microspores appears as a cluster of 4 cells. The microspores are arranged in a cluster of 4 cells. Hence they are called microspore tetrad or pollen tetrad. At this stage, the 4 pollen grains are attached to each other with the help of callose layer. Generally, as the anther matured, the callose layer is disappeared by callase enzyme. Callase enzyme is synthesized by the tapetum. Tapetum is a innermost layer of the anther. The tapetum is the innermost layer of the anther. From the tapetum, callose enzyme is synthesized. The main important functions of the callase enzyme is here degenerate the callose layer. Due to the degenerations of the callose layer, the 4 pollen grains are separated to each other and developed into pollen grains. Totally, how many pollen grains are formed? 4 pollen grains are formed. One more name of the pollen grain is microspore. Here, the microspore mother cell, it undergoes 1 meiotic division to form 4 pollen grain or 4 microspores. The ploidy of the pollen grain is haploid. Next one. Generally, in this way, thousands of the pollen mother cells are developed in the inside of each microsporangium. Later, as the anther matured, these pollen grains are dehisced from the anther. Next one. Here, from that pollen grain, how to develop the male gametes. Here, since totally how many pollen grains are developed? 4 pollen grains are developed. Here, this is one pollen grain. The pollen grain, it undergoes 2 mitotic division to form 2 male gametes. The pollen grain, it undergoes 2 mitotic division to form 2 male gametes. Totally, how many pollen grains are developed? 4 pollen grains. From that 4 pollen grains, how many male gametes are formed? From that 4 pollen grains, totally 8 male gametes are formed. The ploidy of the male gamete is haploid. For the formations of the 8 male gametes, how many mitotic divisions are required? 8 mitotic divisions. 1, 2, 3, 4, 5, 6, 7, 8. 8 mitotic divisions are required for the formations of 8 male gametes. For the formations of the 4 pollen grains, how many spindle fibers are involved? 3 spindle fibers. For the formations of the 4 pollen grains, how many meiotic divisions are involved? 1. For the formations of the 8 male gametes, how many spindle fibers are involved? 8 spindle fibers. For the formations of the 8 male gametes, how many mitotic divisions are involved? 8 mitotic divisions. This is a very important concept. In that microsporogenesis, different types of the problems are also asked in NEET examination as well as the CET examination. Now we have discussed the different problems based on microsporogenesis. This is nothing but one flow chart. This flow chart is represent the entire concept of microsporogenesis. Already we have here the flow chart of microsporogenesis. First of all, the first question is, how many meiotic divisions are required for the formations of 4 pollen grains? For the formations of 4 pollen grains, how many meiotic divisions are required? 1 meiotic division. Second question is, how many meiotic divisions are required for the formations of 100 pollen grains? 1 meiotic division, 4 pollen grains. 100 pollen grains, how many meiotic divisions are required? That's why 100 by 4, 25 meiotic divisions are required for the formations of 100 pollen grains. Third one is, how many meiotic divisions are required for the formations of 8 male gametes? This is a flow chart. In that flow chart, for the formations of 8 male gametes, how many meiotic divisions are required? 1 meiotic division. Next one, fourth problem is, how many meiotic divisions are required for the formations of 100 male gametes? 1 meiotic division, 8 male gametes. For the formations of 100 male gametes, how many meiotic divisions are required? 100 by 8. Approximately 12.5 meiotic divisions are formed, but the meiotic divisions are not considered as a point. Must and should take in a above number, not below number. The above number is 13. That's why 13 meiotic divisions are required for the formations of 100 male gametes. Very simple answers. In examination, the pollen grains are given. Find out the meiotic division. The given number of the pollen grain by 4. In examination, the male gametes are given. Find out the meiotic division. The given male gametes by 8. We will find out the meiotic divisions. The next problem is, how many meiotic and mitotic divisions are required for that formations of 8 male gametes? For the formations of 8 male gametes, how many meiotic divisions and how many mitotic divisions are required? 1 meiotic division, 8 mitotic divisions. This is the fifth question. For the formations of 8 male gametes, how many meiotic and how many mitotic divisions are required? 1 meiotic division and 8 mitotic divisions. Sixth question. How many mitotic divisions are required for the formations of 1 male gamete? 1 male gamete, 1 mitotic division is required. Seventh question. How many mitotic divisions are required for the formations of 100 male gametes? 1 male gamete, 1 mitotic division. 100 male gametes, 100 mitotic divisions are required. The next problem is the ratio between number of meiotic and mitotic divisions are required for formations of 800 male gametes. The ratio between number of meiotic and mitotic divisions are required for formations of 800 male gametes. 1 meiotic division, 8 male gametes are formed. For the formations of 800 male gametes, how many meiotic divisions are required? 800 by 8. 100 meiotic divisions are required for the formations of 800 male gametes. 1 mitotic division, 1 male gamete. For the formations of 800 male gametes, how many mitotic divisions are required? 800 by 1. 800 by 1. 800 mitotic division. The ratio between meiotic and mitotic division for the formations of 800 male gametes, 1:8. This is the 8th problem. The 9th problem is total number of spindle operati formed during the development of 8 male gametes from the microspore mother cell in the angiosperms. Total number of spindle apparatus developed for the formations of 8 male gametes from the microspore mother cell is. This is nothing but the microspore mother cell. The microspore mother cell, it undergoes 1 meiotic division. 4 pollen eggs are formed. In that meiosis division, 2 types of the divisions are present. Meiosis 1 and meiosis 2. In that meiosis 1, 2 cells are formed. 1 spindle fiber is formed. In meiosis 2, 4 cells are formed. 2 spindle fibers are formed. That's why totally how many spindle fibers are involved for the formations of 4 pollen grains? 3 spindle fibers. Each and every pollen grain, it undergoes 2 mitotic division to form 2 male gametes. For the formations of 1 male gamete, 1 spindle fiber is required. Totally how many mitotic divisions are required? 8 mitotic division. For the formations of 8 mitotic division, how many spindle fibers are required? 8 spindle fibers are involved. In meiosis division, 3 spindle fibers. In 8 mitotic division, 8 spindle fibers. Total number of spindle apparatus formed during the development of 8 male gametes from the microspore mother cell in angiosperms. In meiosis 3, in 8 mitotics, 8 spindle fibers. Totally 3 plus 8, 11 spindle fibers are involved for the formations of 8 male gametes from microspore mother cell in angiosperms. These are important problems involved in microsporogenesis. These problems are asked in some of the asked in NEET examination as well as CET examination. Please properly, thoroughly practice these problems.